Hey everyone, it's Ansley and Bailey, and we're going to be talking about our Dutch bucket hydroponic system.
For this experiment, we grew peppers, tomatoes, eggplants, and cucumbers using the Dutch bucket system. And the substrate was comprised of peat, pine bark, perlite, and a hydrofiber lime, along with some other components. We used a drip irrigation to apply the fertilizer, and we used a 15-5-20 nitrogen fertilizer at 2 different rates. We also had intercanopy lighting rods for supplemental lighting and measuring tools to carry out this experiment. The setup was 2 dosatrons, one set for 100 ppm to 24 plants, and the other was set for 200 ppm to the other 24 plants. So 6 of each crops received each treatment throughout the drip irrigation on Monday, Wednesday, and Friday for 5 minutes.
So every week we did a couple things. We trellised the tomatoes and cucumbers as they grew. We took ring clamps and clamped them to the twine that was hanging. And we also picked off suckers and any premature grown flowers on the cucumbers. On Mondays, we measured the canopy. So we measured the height and the width consisted of north, south, and east-west measurements. And then on Wednesdays, we measured the pH and EC using the pour-through method, where we took the leached fertilizer water and measured it with the Grow Line portable meter.
During week 1, we transplanted all of our plants. We did a total of 48, and these were planted in 3 and 5 gallon buckets. The fertilizer at this point had not been applied. And around this time, we also found thrips. This was likely due to a lack of water and excessive lighting. Because of this, we sprayed them with 2 separate treatments, one of Talstar at 6 milliliters a gallon and Kinetic at 0.25 milliliters per gallon. And after we applied these, we saw some burning symptoms from stress.
In week 3, the fertilizer treatments were applied. So we mixed 1,261 grams of Jack's fertilizer into a 5-gallon bucket of water. We had 2 dosatrons pulling from that same bucket. One was set to a 1:100 injector ratio to apply the 100 ppm treatment, and the other one was set to a 1:50 injector ratio to apply the 200 ppm treatment. And again, these were applied through the irrigation scheduling on Monday, Wednesday, Fridays.
At week 4, we saw a decline in our cucumbers. We saw this after spraying for thrips. They didn't recover from the stress that we saw. So we replanted 12 cucumbers with the same cultivar. And at this point, flowers were still being pinched off the plants because they weren't strong enough to support the growing fruit. And by week 5, they were large enough to be trellised and trained with the twine using the ring clamps. And these are images showing before symptoms during that stress.
So we kept our weekly measurements in an Excel document. And so this is kind of how we set up, how we tracked all of that, the width and the height, and then also the pH and EC. We measured 3 plants from each crop from each treatment for measuring the height, and then the pH and EC, we just measured 1 plant from each treatment from each crop.
By week 9, we saw a drought. There was an issue with irrigation, and so plants were experiencing a lot of stress from this. We saw the tomatoes had wilted leaves and the soil was very dry, and images on the right show those leaves being really wilted. We also began to notice the nutrient mobilization at this time. We saw that the 100 parts per million treated crops had more flowers and fruit development, and also yellow leaves in the bottoms of the tomatoes and eggplants, while the 200 ppm treated crops had a denser foliage at the base, but they had less flower and fruit development.
So we finally got to harvest some cucumbers in week 10. They were the first crop to be able to harvest, and they were the only crop that produced continually so that we could track the harvest. Everything else was only harvested once.
And at week 13, we ended the experiment. So we harvested everything that we had just to be able to measure all of the yield. So we harvested all the fruit from peppers, tomatoes, and cucumbers at the end of the study. No eggplants were mature enough to harvest, so we didn't harvest any of those. All fruit was weighed, including 1 destroyed plant from each crop from both treatments, so we could measure the fresh and dry weight to compare biomass.
So now to our results and conclusions. So here we have the cumulative yield of the crops that were harvested. We can see that the 200 ppm produced a little bit more fruit in the cucumbers, but not in the tomatoes or the peppers. The 100 ppm crops actually produced more peppers and tomatoes.
So data collected measuring the height and width shows the plants receiving 200 ppm grew faster than the 100 ppm. With the 200, there was a drastic spike for cucumbers around week 7, maybe. And so that's recorded in this chart here. But overall, they had both a pretty steady incline and increase in growth, but the 200 ppm had overall a larger canopy size.
Here we have our pH and EC. Relatively throughout the experiment, the pH and EC measurement stayed between the desired ranges for all the crops. There was a little bit of increase and then a decrease at the end, especially for pH. It kind of dropped off towards the end. And then with our EC, the 200 ppm showed a lot more fluctuation, but also there's kind of that little bit of a decrease towards the end as the plants are using more nutrients to produce those flowers and fruits.
And for these charts, we have the fresh and dry weight. So you can see the 100 ppm plants compared to the 200. And so overall, the 200 ppm plants had a higher biomass. The cucumbers are the only exception, though. Both treatments had relatively the same biomass weight for both fresh and dry. So the higher nitrogen amount applied to the plants created a higher biomass weight in the end.
And there we have it.
Thank you.